Good morning. Hi there. Okay, so we continue our seminars of coffee breaks with TRANSFAC. And today we start a new form, a new type of seminars, coffee breaks, a bit more intensive, I would say. So we are inviting developers of software which is using TRANSFAC. And we'll start today with a software which is called Match Suite, which is a part of our suite of software we provide you to use TRANSFAC to predict your binding sites. And I invited Daria Stelmashenko, who is a main designer and developer. Actually, her team is developing Match Suite, and she's introducing today this software, showing you like a software demo. So basically, we will go through all features of software. You'll see how to use it and which button to click. And also, as usual, don't hesitate to just interrupt us and ask your questions. All right, so now, Daria, the floor is yours. You can share your screen and start your presentation.
Thank you, Alexander. Hello, everyone. Yes, so let's start. So I'll start from the very beginning, from what you see once you log in to our portal. Commonly, you would either see TRANSFAC if you're subscribed to TRANSFAC only, or you have TRANSFAC pathways. Then you also see that TransPath and human PSD component integrated to it. And in both cases, as you can see here, Match Suite is an independent point on which you can click and immediately actually get inside the software. But I would like to start maybe from the interface that is more usual to you, from the database itself, and show you how Match Suite can be accessed from the search results inside the database. For this demo, I'll use the extended build that includes TransPath and human PSD, but almost everything that I'll be showing you can also see from the TRANSFAC itself, except from some components that I'll comment on additionally that they are not included in the TRANSFAC native build. So that is the search interface you perhaps know already. And Match Suite can be used in both modes for single gene analysis and gene set analysis. So for a beginning, my suggestion is to just search for a single gene in the database interface and have a look at this report and see how Match Suite can be launched on it. So for the demo, for instance, we can use the EGFR gene. Click on search. For sure, the search will produce different results for various organisms. Very important message is that Match Suite can be launched only on human genes. It was designed to work with human genes only. So in our case, we would be interested to look into EGFR for Homo sapiens. You can actually launch Match Suite already from here, simply by ticking this gene and clicking here on Match Suite single gene analysis. But prior to doing that, maybe just as a short introduction to the topic itself, just to remind you about what are transcription factors, what are their binding orders, what are the matrices, and how actually the genes are regulated, let's jump into the locus report of the EGFR gene, have an overview of it, and have a quick view into its transcriptional regulation section. So any locus report in our database, as you might know, starts from several sections, and you can find the table of contents of the whole report here on the left. And as I was promising, I told you that components that differ for TRANSFAC native and TRANSFAC pathways I will mention. So for instance, you would not see the pathways and interactions or and drug annotations and biomarkers information if you're in a TRANSFAC build, and you have an advantage of seeing this information when you're inside the full database mode. So if we skip introduction and gene ontology section, biomarker, which is available under human PSD, as I mentioned, you'll end up in the gene section. And scrolling down, you'll be able to, okay, this is also interesting. So this is the expression or profiles of this gene, and we will talk a lot about expression today because it is one of important inputs that Match Suite is working with. But nonetheless, it's not in the focus yet, but maybe just to show you that in principle, EGFR, since we'll be analyzing it today, is highly expressed in placenta, and then the tissues decrease. In our examples today, we'll be looking at a liver and at a breast tissues as during the studies of EGFR, but you can see that it is also expressed in a number of other tissues and less expressed in others. Okay, but we were about to look at the transcriptional regulation section of EGFR as like an introduction to what we're going to be doing today with the match suite. And here in this browser view, you can actually click on the fragments, see the there's experimentally validated, not predicted sites, and you can click on any site for further information. For instance, here, if I click on the EGR, I can see the details, and I can go even to the site report with further experimental details about it. And inside it, in just a second, yeah, you see additional information about the binding site, the sequence, and manufacturers, promoters, and so on. And you can also see the matrices associated with this binding site. And if you click on the matrix, you'll jump to the matrix report. And from here, perhaps it would be more obvious what are the matrices at all, because we'll be talking about them a lot today also, because this is something that Mesh Suite is also predicting. So this
I can maybe I can just add here.
Sure.
Yeah, sure. Yeah, just sorry for interruption. Just to just returning back to a bit to what we were talking about before. So basically, but maybe some of you were not in the previous sections. So if you show these matrices again, Daria, so you see this actually representation of the same matrix in two different ways, like on which strand it's presented, on the plus strand, on the minus strand. So basically it's the same matrix. And for each nucleotide, you see basically the number of times this nucleotide was observed in that motif of the binding site for that matrix. And how these numbers come up, so if you scroll down, you see basically how we constructed that matrix. So it was constructed by our team. We collected known binding sites for EGR1 transcription factor from various vertebrate organisms. So it's actually summing up all functionality of those factors in different organisms. So they have a very similar, almost identical DNA binding domain. So the binding of those factors to the places is very similar in different organisms. That's why we can collect them and align them and build one positional weight matrix from those sites all together. And you see more information about structure of those factors in different organisms. And also very important, you see other matrices which are actually connected to this site. So first, these two matrices actually were constructed using that site itself, but there are other like 23 matrices in TRANSFAC database which are collected from various data sources, like from ChIP-seq experiments, from bacterial one hybrid system, from other databases, basically from SmileSeq, so from various methods. So where these factors, these family of factors, and basically in this case, we are talking about EGFR1 only, from in vivo and in vitro experiments. And you see similarity between these matrices on one side, but also a little bit difference between them, which reflects the different binding specificity of those factors, although the factor is the same, but depending on the condition, depending on the environment, on cell lines, or on the situation, on the tissue, the binding specificity might be slightly different. And that's how we document that all in TRANSFAC. And that's very important to understand, that we actually collect all the matrices which you found in literature or in the databases we are not really throwing them away or building one matrix and keeping it like a non-redundant one, but really collecting all these matrices all together and allowing our algorithms to find the one which fits the best to the conditions we are analyzing. Okay,
yes, this we will talk a lot
a little bit
about
of
today.
introduction to the matrices again.
Yes, thank you very much, Alexander. Just to mention, so that particular matrix was built based on 194 references, so quite a lot. Okay, if we now give you a brief introduction, just to remind you what are the matrices, these binding motifs, self-transcription factor binding sites, that there can be many of them. We can now open Match Suite, I believe, and start with this EGFR gene analysis. So you can click here, analyze EGFR with Match Suite. It is like more obvious, but just to remind you, in our search results from the very beginning, I mean, once you have entered the database and have found any gene of your interest, you could have also launched Match Suite simply by selecting this gene and clicking on Match Suite single gene. So it doesn't matter where you click, it will do the same. A transmission from the portal interface to the Match Suite interface will happen now. So we're now in Match Suite. The system has identified the ensemble ID of EGFR, and now we're ready for launching the analysis. So this is how the analysis launch interface looks like. I'll zoom in a bit. Please let me know in case further zoom in or zoom out should be applied, or if you see well, let's proceed. So first of all, we can specify the name of our launch. So since I already have an EGFR results pre-calculated here, it automatically put a number 1. It can be like test 2 or whatever. Now more important, so the tissue. As we said, Match Suite is intended for analysis of human genes in the conditions that you specify. So one of the conditions that you can specify for this analysis launch is the tissue of your interest. There are 61 tissues in this list supported so far by Match Suite. I'm not sure. Okay, we can use any tissue because it's just a launch to show you how to do it. But you can immediately see actually in which tissues your gene of interest is expressed. If you have not checked it before on the portal, you can click here. And Match Suite will show you the expression patterns of this gene across 61 supported tissues. Just give it a moment to compile this diagram from the table data that it has. And by the way, all gene expression data that we have here is taken from the Protein Atlas, just for your information. So again, we can sort by the expression value in various tissues. And as I said, today we'll be looking at the breast and liver examples. But this is further on. So for now, it's just how to select the tissue in the interface. What will tissue selection affect during the analysis? We'll discuss it while we'll be looking into the pipeline underlying the Match Suite. But even here from the very beginning, you see the note that it's Phantom 5 database, perhaps someone knows it. So we're taking the tissue-specific promoters for the selected tissue for studying and the gene of your interest when you select the tissue. That's one of the ways how we use the tissue. Then the promoter range, which is by default from -500 to +100 relative to TSS, can be changed to any custom range of your interest. So you might have noticed here the maximum is 2,500, sorry, and I believe up to 500. Oh, so okay, up to 1,000 can be specified. Okay, you can do it if you want. You can enhance it or make it smaller depending on your preference and how wide the window for scanning should be. The single gene analysis, in addition to the promoter range, which you define here by the tissue and by the range itself, will also be taking into account the known enhancer and silencer regions of the gene of your interest. And if you have specified a tissue, it will of course select only those enhancers that are known to be active in the tissue of your selection. But if you have not specified the tissue, and EGFR is a good example for that because it has a lot of known enhancers, and we will see it in a moment in the results observation, it will take all of these regions into further analysis for a search for transcription factor binding sites. Next, you can select the promoter type you would like to study, most 3', 5', on the best supported one. And perhaps this is also an interesting feature. You can immediately here optimize the profile. Actually, it's very interesting, and we will talk about this a lot if you have ever used match searches on the portal, you might have noticed that they're always very quick, and they're very good, of course, but they're always using the standard profile. So the matrices which are used for the site search are always the same collection of matrices. And here in Match Suite, and we will see it, actually the profile is constructed based on the input. But even here, from the launch window, you can already optimize this profile by selecting geoterms we have found to be interesting. So these are not all geoterms, of course. It is the geoterms that we have pre-selected, and we have found them interesting for profile optimization, and of course, in terms of the profile consistency after applying them. So you can select any process of your interest from this list. And then what will happen is that while performing the search for transcription factor binding sites, only matrices belonging to transcription factors whose genes are belonging to the selected GO terms will be considered. So this will be a very strict selection. You will not find any other matrices but those which have connected transcription factors whose genes are known to be categorized as the selected GO terms. And now we are ready to click on the next button and review our input parameters as a summary of this launch. And if everything is okay, if not, you can go back and reselect it. We can click on launch the analysis. So just to show you that analysis can be launched, I'll click on it, but we will not wait for this analysis to finish online. Instead, we will now jump into the already pre-calculated results that I have prepared for the demo. So let me please stop this analysis. But okay, maybe before I stop, let's have a look. Here you can view the input parameters if you forgot. This will show you on which parameters the analysis was launched. And you'll see the progress of the analysis in percentage, but anyway, let me stop it since we do not want it actually. And you can, by the way, delete the unneeded results. You can just say okay and remove it. And now let me show you how it would have been if we have waited for the analysis to complete in case it was just EGFR gene with any additional specifications. So no tissue selected, no geo categories for profile optimization, just EGFR gene as it is. Basically everything that we know about EGFR was used for this analysis launch. So let's view these results. When you click on View the Results button, you now will immediately see the results interactive visualization. And I believe that if you have never seen Match Suite before, this looks a bit frightening because there's a lot of different colors, numbers, question marks. Well, the good is that each question mark actually has an info box behind. You just wait for it to appear, and it will immediately tell you what is actually displayed. But of course, still you have spent a while to look into that and understand. And for that reason, my suggestion is to first go through the analysis report to talk about what was the analysis that was performed, what were the steps, what were the tables that were constructed, and then we will go to this online visualization with full knowledge of what you actually see here and how it can be now fine-tuned with various filtering in the interface. So my suggestion is to start from the analysis report. Okay, you see the brief summary. So who has launched this analysis, when? Then, of course, you know what was the input. So our input was the EGFR gene. All actual input parameters are summarized here. You see the promoter region in focus. You know that no tissue was specified and no geo categories optimization was used, since it is not mentioned here. And at the very beginning, the system is already giving you information about the key transcription factors which were predicted to be regulating the input gene in the specified conditions. But before we look into this transcription factors and into the values and the columns of this table, perhaps I would like to still provide you a brief overview of how the analysis is looking like in the system. And when you're going to be running your analysis by yourself, you should know that actually Match Suite is very much self-explanatory. First of all, each analysis report from the very beginning has this link to the so-called methods section. And if you click on this methods section, it will be methods for single gene, since we are now analyzing the single gene, you'll find a very, very comprehensive description of how this analysis is performed. So don't be afraid if today we are not going into the very, very deep details. We will go if you would like, but if not, and then if after the webinar something comes into your mind and you forgot to ask it, be sure that you'll find it in the methods. Here, absolutely everything with all the formulas, the links to the geneXplain methods, which were applied for running this analysis, is shown, explained in all the details, and how the filtering was done, how selection was performed, everything, how to read those results, and so on. So be sure to find it in the methods section of the analysis report. And there's one more important help file that you might want to have a look at. It's the user guide. And the user guide is split into the gene set and the single gene analysis. So in our case, it is a single gene analysis. And it starts, as you can see, from the BCal interface, exactly what we were doing, how you find the gene, how you launch it, and so on, and how then you launch the analysis, which we were just doing. But in addition to that, it will be also helpful for you to look at additional explanations of what can be found on the interface, how to interpret the results that you see. Everything that I said that's looking very complicated, and we'll, of course, look into it today together, is described here by text, graphics, and so you can be sure to find all questions, all answers to your questions here. Of course, today we have an advantage of the live discussion if any questions arise. But as I said, if after the webinar you realize that you have missed something or forgot, you're very much welcome to refer to the user guide and to the methods. Okay, now returning back to the report, if you scroll down a bit, and as I said, this table needs to be first understood how it's constructed at all, you'll find the overview of how the pipeline looks like. I have actually prepared a very short slide that would help us to go through this pipeline quicker. So we start from the input gene right here. As I said, what are the regulatory regions? So it is the promoter that we select either for the tissue of your interest or just the ensemble promoter. You select whether it's a 5', 3', the best supported promoter. And then the so-called affinity match is a method in our platform. It is clickable, as you have just seen in the methods. You can open it and read more about it, how this method is working. There's the publication cited there. And so for this very first run, which is important, we're using basically the full profile for the vertebrates. So we're using a lot of matrices, 5,763 matrices I used for this launch. And then, as a result of this launch, this method is reporting us the scores, the affinity scores of the transcription of the binding sites and the p-values of the positional weight matrices for each of the regulatory regions. So the enhancer, silences, and promoter of the input gene. And
let
yes?
me maybe comment here about this affinity.
Sure.
Just a very short, because of course you can read it in the method section, and there some very good description. It's a new method. No, but please stay with the.
Actually, I want to just since you're talking about it, I'll just open it for your convenience. So you
All right. So actually, the affinity score is something very special. So what happens? You know, this. Of course, we are working with individual matches of the transcription factors. So like these small sites of whatever 10, 12, 20 nucleotide long, and the score of binding of the factor to this position is calculated by the match score, the usual match score, which is described and published in many papers. But to select first which are the most interesting factors to look at, we actually score the whole promoter, or actually not only promoters, but also enhancers. So like in this case, and you'll see the EGFR has not only one promoter, but it has several enhancers. And then in each of that region, we basically scan the matrix, each matrix of these 5,000 something matrix from TRANSFAC, and kind of build a curve of the score across the whole sequence. And then there is a certain way of summing it up. and calculating overall affinity score of the binding of this factor to the whole promoter, not just. And of course, if you have just one side, but very high ranking, very high scoring, it will be a high score, but you can also get a very high affinity score by having many of low scoring binding sites. So basically, it again calculates the affinity probability. You can say it this way, that these factors actually regulating binding the whole promoter and regulating the gene. And of course, we calculate also p-value of that score to be able to filter it and to put some cutoffs. So that's what I would say. You're showing this?
Yes, I'm showing the method in the system.
Right.
Also, you'll find in the references the link to the publication, quite recent one from 2020, where this method was first applied, actually, and then it was implemented by us in the Mesh suite. And of course, it is also contained as a reference in the methods section of the Mesh suite, so be sure to find it there in an appropriate place with all the description. Okay, let's then proceed further to the brief overview of the applied analysis. So having applied this affinity match on tracks, we then apply the filter by the affinity p-value. And for each of the matrices in the resulting table, we select the one with the best affinity p-value among all regulatory regions. Okay, then so-called orthologous and paralogous extension and redundancy filter is applied. That is actually a very interesting step. I also have a table open for it, but it looks quite complicated. So maybe first let's talk about it just by words. So to make it simple, when we make orthologous extension, it means that if a matrix in our database is actually linked to a mouse transcription factor, it can be also used to identify the transcription factor binding sites of its orthologous human transcription factor. So it can be used because this binding modus are very conservative among different species. And for the paralogous extension, as an example, so if we have a matrix linked to a human transcription factor, which is closely related to transcription factors with nearly identical DNA binding domains, then these paralogs can also be assigned to the hits of this matrix. So we're actually extending. And you can ask why. I mean, you already have a lot of predictions. If you ever saw how match output is looking like, it already is predicting a lot. So why are we actually even extending it more? Because then we're applying the redundancy filter. And the redundancy filter is actually fine-tuning the results to the conditions which you study. For instance, so here in this case, this redundancy filter will select. So okay, and it is orthologous and paralogous extension was summarized by Alexander and Professor Edgar Wingender in a so-called factor cluster table, which is of course also linked to the methods of Mesh suite, and you can open and have a look at it. And for these clusters they have introduced, we select only one matrix for each cluster. That is minimizing the affinity p-value. And at the same time, of course, this expression profile is taken into account. So actually, you select the best matrix for the conditions that we're studying. And this is what this step is doing. And then this filtered and optimized list of matrices is actually used to construct a new profile. So if we have started from a huge profile of basically all vertebrate matrices, now we have an optimized profile that is fine-tuned for your input gene and for the conditions that you have specified at the input. Actually, this is a unique feature of Mesh suite. No other match tool implemented by us is doing it. Only Mesh suite is really generating an optimized profile. And again, so what is a profile? It is a collection of positional weight matrices that will be used for the search for transcription factor binding sites. So it's actually the main. So this is like the very valuable input for the next match run, which will be performed, because it will be fine tuning your results. And so this optimized profile is used for launching our classical match on tracks method in the platform. And then the output of this analysis gives you the list of matrices, which are then eventually mapped to transcription factors corresponding to them. And this list of the transcription factors is then reported in the report. And while selecting, okay, so here I have a note. Sorry, I missed it here. So what the matrix table actually gets as a result of this match run are two important scores. So number of sites, of course, number of binding sites that were found for each matrix in the studied regulatory region, and the site score. So we'll take the best score among all sites of the respective matrix for further consideration. And once you have constructed this matrix table and constructed the factors table just based on all transcription factors referring to these matrices, you should then actually select which factors are really regulating your input gene in the given conditions. And for this, we're applying the ranking procedure. So we are ranking these factors. We have different ranks applied, listed here. So the main one, I believe, is the minimum affinity p-value coming from the best matrix of the factor. And then, as I said, expression information is taken into account by match suite. So that's why it is basing its rank either on the factor expression in the selected tissue or on average value of factor expression among all supported tissues, and the rank of the factor expression either in the selected tissue or on the average of its, the rank of its average expression among all tissues. And once you apply the rank sum to the factors corresponding to the eventually selected matrices, you get the eventual result, which is then making it to the report. And now when you know what is in the report, let's actually go back to this report and start from the very beginning. So this is exactly the table of transcription factors which was constructed based on this ranking. So you see it's sorted by the rank. And so what you see here, you see the factor name, which is clickable, and it will take you back to the portal interface to the respective locus report of this gene. You see the respective gene symbol, which will then open. You see the transcription factor classification, the TF class classification introduced by Professor Edgar Wingender, and it's clickable also. It will take you to the classification table and see all these families of Fabulous information. You see the site model, so basically the matrix from which this factor was derived. And then you see the values. So you see, our goal was to have some kind of score. That's why the score actually is the minus log from affinity p-value. And as I said, you see the rank. The ranking, we remember, it was a rank sum by a number of parameters from the slide. And then you see since we have not specified any tissue, those values are given for the average expression values of this transcription factor across all 61 supported tissues, and its specificity. So actually specificity, especially, it's easy to see from the online interface, that the more reddish the color is, the more specifically this factor is actually expressed in some particular tissue. Like here you see red, that means that perhaps there's only one or two tissues where it is highly expressed, and for the rest it's much less. And yes.
Sorry, so let me maybe summarize as I understand. So it was very complex schema how this algorithm works, but actually in summary, as I understand, what we are doing, so you have your promoter, you have your one gene, and you want to know which transcription factors most probably are regulating my gene in that particular tissue, right? So I have the set of matrices from TRANSFAC, which is very big, like 5,000, almost 6,000 matrices. And actually the match suite in this option allows you to select which of the matrix. First, it selects which matrices out of this big collection as most relevant. And then on the second step, to select which factor connected to this matrix is most relevant. Then you get a small list of factors which are algorithm believes that's most relevant and most important, and most you get most of the evidences allowing to predict that these factors are actually regulating my gene in this particular condition. And actually in that table, what is interesting to see here is that for instance on the line 3 you see this factor is TCF7, but the motif for this factor is recognized by the matrix called TCF1. So which is really clearly showing you this paralogous extension when the motif which was built by the factor binding sites from TCF1 can be extended towards the predicting binding sites to the paralogs, like also TCF7 and other paralogous factors which have the very similar DNA binding domain. So that's how the paralogous extension works.
Mm-hmm. Thank you, Alexander. Yes, please stop me if I become too technical. No. Okay,
no, just,
uh-huh.
giving some comments.
Thank you. Sure. Okay. So yes, and by the way, you see it had 34 enhancers and 0. So that's really a lot. I mean, in the general setup, when we have not specified any tissue, that's a lot. That's what we're now looking at. Now this results details with the schema we have already gone through. So the steps with all the, you should also know it. So all these tables and steps written here, it's not just for beauty, it's because they're actually all clickable further in the report, and you can have a look at all intermediate tables. You can see how the profile was constructed, how matrices were looking at, like before filtering, after assigning all transcription factors, after applying the rank sum to them, and so on. So all the steps here are clickable. And you can click on, okay, let's click on any supplementary table, for instance, and you can see then on each step of analysis how the intermediate result is looking like. It's not as nice and as colorful as the resulting table, but it is of course very much informative and gives you an idea of how the system was proceeding through the steps. So the steps we have discussed already while looking at the slide. And here are the matrices. They're here below in the report because the factors are like the main result. But as you know, in the analysis logic, we first found the matrices, and only then we were selecting the factors that are corresponding to them. So for each matrix, as we know, we see the number of sites found, the overall numbers, the affinity score, the minus log affinity p-value, and the site score which was assigned. So it's the best site score which is taken for each matrix. So after the matrices summary table, you see actually the summary of your regulatory regions that were submitted to the analysis. So in addition to the EGFR promoter, as we know, all enhancers were taken because the tissue was not specified. And you see here the total number of sites which was found for each of the regions. And then for each individual matrix and associated factor, you see the number of hits. So you see the number of binding sites found on the intersection. And in the interface, this is all nicely colored. We'll see in a moment. And then you see the references with all the links and how to cite the software and the disclaimer. So this is how the report looks like. And now when you already know what are the actual results, we are ready to go here to this page with a lot of colors. So let's first of all have a brief overview of what you see here. So this is what opens immediately if you open on visualize the analysis results. This is what the system will open by default. I think I should zoom out a bit because otherwise it's hard to have a look at it. So here on the left, you see the tables of factor view by default. This is our resulting table, absolutely the same as we just saw at the very top of the report. And this is our matrix table, absolutely the same as we have just seen at the bottom of the report. As you can see, additional filters can be applied. We'll return to them in a moment, but just for you to have an idea of what you see on the screen. So it's the factor view or the matrix view in the left upper rectangle. Here, at the right upper rectangle, you see all the regulatory regions which were submitted to the analysis. So in addition to the promoter, we see all those enormous amount of enhancers that EGFR gene appeared to have. And as I said, here in the interface, by color, the more intense the color is, the more sites were found for the respective matrix in the respective regulatory region. And so all found matrices and associated transcription factors written below them can be found here with respective values of the numbers of found sites, and the total number of sites is then reflected in this very first column. These are, of course, clickable. So this would take you back to the EGFR locus report that we have been already looking at today, but let me just show you that you can open it once again. And by the way, in this report, I think that we did not do it at the beginning. You can, of course, also find all this enhancer and silencer entries in the database format. And here you have them in the format of the table. Next, so each enhancer report can be accessed. You can also click on it and see information on this enhancer. Where was it identified? In which tissue is it active? And so we have the overview with the interactive image, the features, identifiers, and links to the publications from where this information was extracted. So this is all clickable from the interface. And at the bottom, you see the genome browser. And if you click on any regulatory region, for instance, now I'm clicking on the promoter, you will immediately be taken in this genome browser to the beginning of this regulatory region. And here you'll see the found sites, actually something else in addition. We'll have a look in a moment just to show you that I'm clicking on different regulatory regions, and immediately the image here is changing because it is jumping to the beginning of respective regulatory region. And to go into more details on the genome browser, actually, there's more that can be done here. You can select the tracks that you can see. So by default, you see the track of genes, you see the track of found sites, and you see the promoter and enhancer tracks. You can also view all enhancers, all variations information, or experimentally validated transcription factor binding sites, or conservative regions of genome. Let's do it on another example where less binding sites would be found, simply because we will apply more conditions to the tissue, geoterms, and so on, because on such a big number of sites, we will now get lost in all these visualizations. But just for you to know, you can just tick all these sites, and they immediately will be displayed. Like here, you immediately see the conservative region of the genome, and you can compare where your binding sites, which were predicted, are located. You can
Just
also apply,
maybe
yes?
one short comment here concerning the conservative regions, how important they are. You see that very often they are located. It's a conserved region between various big set of vertebrate genomes. And of course, you know, the protein regions are quite conserved, but here is most focused is on non-coding regions. Like in this case, you see it's located in front of the promoter, in front of the start of transcription of EGFR gene. There are a number of conserved regions, and they cover the actually most number of found sites. And you know, actually, the role of conserved regions is really sometimes really to identify those transcription factor binding sites which are behave like the first comers. So those you know, they will sometimes called as pioneering factor. They are coming to open the chromatin, and they are always the same in all across the you know, several species, similar species, and that's why they are located in a conserved region. But that's not all the factors and not all the binding sites which are actually acting on promoters. If you shouldn't be you know, really too restrictive, just looking in those sites which are just conserved and forgetting about other sites, because in reality the regulation of gene is done by many other sites which are actually not conserved. Yeah, just a little comment.
Thank you very much, Oksana. So actually, you now saw overall interface of how, in principle, single gene analysis results look like. But just to remind you, we were analyzing it, EGFR gene without any restrictions. And for further demo, I would like to move to another example where actually additional restrictions were applied, and which is more meaningful, I mean, in terms of what we'll be looking at. For instance, here's a run referring to EGFR gene in liver tissue. I, again, did not apply any gene ontology restrictions to this run. I simply selected the tissue as liver. And you can immediately see that I have only one enhancer left. So it's not already the huge list of all known enhancer regions of this gene. So there's only one enhancer assigned to liver tissue, known to be connected with EGFR. And okay, so now you can also see the total number of sites. I would like to draw your attention to it because now we'll start applying various filters and these numbers will change. And of course, the number of visualized sites will also be changing proportionally. So as I have already briefly shown you, on the matrix view table, there are two filters at the top. which you can apply. So you can ask the site score to be more significant, but actually here it the scale itself is adaptive depending on your output. It's not, that it's always like from 0 to 1. It is always different depending on the actual values that you have in your results. So here we already see that the site score is very good. So it's starting from 0.8. Yeah, it could be starting from below threshold depending on the output. But here again, you can request it to become more. So while applying this filter in the matrix view, at first you see that the only thing that is changing is actually here in this table where I'm working. And once I like the result, like I had 22 entries when the score threshold is 0.9, and I have only 2 entries when it's 1.0. And if I like what I see now, if I actually want to now apply only those 2 matrices that are left after this filter to the overall picture of results that I have to click on Apply Changes button and wait a moment. And you see, instead of this 186 and 19, we for sure will now see a different number of sites because the number of matrices has decreased significantly. And here, this long list of matrices will be now consisting only of 2 matrices. So everything refreshed, and on the genome browser view as well, you see only the sites corresponding to those matrices which were left after applying the filters. And similarly, you can work with -log affinity p-value. So first of all, we can remove all filters, return to the initial calculations, return to the matrix view, and apply the -log affinity p-value. And now I immediately get only 6 matrices. And if I like it, again, I can apply the changes. And my results will be recalculated in the promoter and enhancer regions and in the genome browser. Just give it a moment to apply those matrices. So that's how the filtering works. Filters can also be applied from the genome browser. Like here by default, there's no filter. Okay, somehow, I'm sorry, I think that it is something with my cookies today. Okay, my genome browser is looking not very well, but anyway, so I can view only sites near the conservative regions of the genome, for instance. And that would leave me with a selection of sites that were located near the conservative regions, which you see here. So only the sites that are intersecting with those regions will be displayed then. Okay, let me perhaps remove all filters once again. And here I'm again at the promoter entry, and I can also view the experimentally validated transcription factor binding sites if I want to have an idea which of those predictions were previously supported by experiments. I can zoom in the browser to make it look a bit better. So for instance, here you see this EGR and NGR, and here in the results, I see those EGRs as well. And if I click on the entry of any site, perhaps I should have done it before, sorry for not mentioning it, I'll see all information about this site. I'll see the sequence, its position, the properties with the scores, and I'll see the site model. And if I click on it, I will immediately end up in the entry of the respective site in our database. We have been looking at one already today with all the supported information behind and the consensus view. So that's how the Alexander, you want to add something, right?
Oh, yeah, right. So basically, as you can see, we predicted many sites and not all of them actually matching the known binding sites and documented in the TRANSFAC database. We found these 2, and that's interesting point because actually what we documented TRANSFAC are all binding sites found in very different conditions which can be regulating EGFR gene. And but here, what we are doing when we are predicting these binding sites, we are putting a particular condition. We are putting a condition on liver. So these binding sites which you see and predicted, they are in respect to liver, which means not all those known sites which are documented in TRANSFAC, they reflect regulation of EGFR in liver. That's why they might be not predicted here. So that's important. And concerning this EGR1 factor, it's it's very much related to the regulation of this gene, because it's epidermis growth factor receptor, and this EGR1 is a factor which regulating this gene in many different conditions, and specifically in liver. And if you click on it on that site, you may see which conditions there were where this site was found, which was documented and then put in TRANSFAC. So as you see, and also important to understand that all known sites which are collected in TRANSFAC, which is quite a lot, still describing only a very little fraction of those those sites which actually can act in the genome to regulate my gene. So that's very important notion, because which tells you that without predictions, you really miss a lot. If you just based on literature information of those sites which are known and identified so far, you may lose a lot of predictions, a lot of facts of real regulation of your gene by a particular factor. So you have to do predictions.
Mm-hmm. Thank you very much, Alexander. Just another very quick point I wanted to show in this tables that we look at, we can use them as interactive tables for sorting, right? So I can sort like by the number of sites, by minus log affinity p-value. And if I forgot what were the best metrics, so to say, you have this button, best matrices on top. So the best is according to this rank sum we were discussing before, but so in principle, in the interface, when you play around with the values, if you feel that you have lost something, you again bring best factors on top, and here you are with the correct, so to say, sorting with the best ones listed, first ones. Also about this color, I told you that from the interface, it's much more obvious. As I told you, so green means that basically this factor is generally evenly expressed among all tissues, and red means it is specifically expressed in some particular tissue, and you can immediately check it. I mean, you see red here. If we click on the respective entry in our portal and refer to the yes, exactly, refer to the expression profile, you can immediately see that its expression profile is very specific, actually, and you can expect it different for that one, for instance, because you see that it is not yeah, so you see that it's not specific for some particular tissue. However, in liver that we are studying, as you see, it's number one tissue where it's expressed. So that's why you see it's ranked 1 out of 62. Okay, and this is the deviation to average. So this is how different the value of expression is in liver of this particular gene referring to this particular transcription factor, how different it is from the general expression value across all tissues, the average value. So this is the score difference. Okay, so this is for a single gene analysis overview. I want to show you one more example when not only the tissue, but also the geo-ontology term was applied. So inflammatory response was selected by me. Since it's the same gene, and you already know that in the original run, when there were no restrictions, there were a lot of enhancers. For liver, it was very specific with just one enhancer. And here for the breast, you see that there are 4 known enhancers for EGFR. Of course, the results look, I mean, technically they look similar, but they're absolutely different. And if you start comparing the reports produced by the systems for each of those cases, you'll realize that they differ very much. Of course, there will be some similarities because we're talking about the regulation of the same gene, but due to the difference in the conditions that you have specified upon the analysis launch, the results are actually different. And you have to expect that, and it is actually the advantage of the system, that it is sensitive to the input conditions. I think that from the overview point of views, I mean, how the interface is looking like, how the report is looking like, the very brief explanation of the analysis schema, I am done. And I would like to answer any questions that you have. in regard to the single gene analysis in Match Suite. And if not questions, maybe if you want, if you're interested in some particular gene, we can make an analysis launch, and we'll proceed to the gene set analysis at the, later on, and then stop and see what would be the results for the genes of your interest. So if any one of you is interested to look at the results of Match Suite run over the gene of your interest, please just type us the gene symbol in the chat, and we will make the respective launch. Or any questions are very welcome. If something was not clear, if you want to look into some for further details of
All right, so yes, please put your questions in Q&A, and also, by the way, put your likes if you are watching it in YouTube. So that's very important for us. And you also, if you have your beloved genes of your interest, so just type the gene ID here in the chat or even in Q&A, and we will launch it, and we'll see the result at the end of the presentation. So give us a gene, but we'll see when you put it in the chat. But for now, maybe you Daria can continue with the second set.
Okay, we just have a gene request. So it's P53 that we
all
received.
Right, so we have a request. Okay.
Yes, we have a P53 gene request. Thank you very much for this. But while we were requesting you to type on the gene names, I remembered one more important feature I forgot to talk about. When you have applied the filters to your tables and you like how they look like and they recalculated in a nice way, and you want to save these results, because of course, it's, as I said, everything is being recalculated when you click on the apply changes. And so just a moment, let it finish, since I applied the filter. So the table of regulatory regions is recalculated, the factor table is recalculated, and the matrix table is recalculated. If you want to save them, just click here on download tables, and you will save those filtered results. So they will be saved in a zip archive, all 3 tables, and then you can work with them further in any way you would like. So this filtered tables can be saved. Otherwise, in the interface, by default, each time the default view will open with the tables that the system has calculated. Okay, so thank you very much for sending us the gene to proceed with. I'll start it on another account, not to disrupt our demonstration now. So, by the way, sorry for that, and many thanks. We didn't show how to launch the analysis from Match Suite interface. So first of all, if you log in to Match Suite from the portal directly, like I showed you, there's a Match Suite here in the product list. And if you just click on Match Suite, this is where you will end up. And you have 2 options, either to view your previous launches, and this is what we already did today. You see the input parameters that were used for the launch, and you can view the results or delete this launch, or you can start a new analysis. So in case of single gene analysis, I click on Next. Here I can type either ensemble ID or entrance ID or the gene symbol. So I'll type the gene symbol. It will be P53.
Well, it should be TP53.
Okay, thank you, Alexander. This is why I like biology knowledge. Okay, so TP53. Next, let's give it a while for the system to identify the gene. Okay. Do you want to do it in any particular tissue? Or
so do we have any wish of a tissue?
Like I can give you an idea about
brain. He wants to do it in brain.
Brain. So in brain, I think that for brain we do not have brain, but we have
amygdala, put it in cortex, maybe.
Or cortex. Yes. Yeah, so yes, cerebral cortex. Okay, perfect. So we can use the default promoter range, or okay, we can use another one. So for you to see that it is really changing, like I can use like minus 700 to 50. Let's keep the best supported promoter. I'm not sure if you want to optimize the profile by geo terms. You just saw an example where it was optimized. Perhaps let's not do it, not to restrict our search. We have already specified the gene and the tissue type. Okay, so yeah, the question is already answered. And we can click on Next, agree with the input parameters we have specified, and click on the launch analysis. And now let's wait for this run to finish. And while it will be finishing, we can go back to the window. Okay, let me close. I have a lot of tabs that we do not actually need, right? Let me find the window where we were working. Yeah, I believe it is that one. And we can proceed to the gene set analysis. If there are no further questions, I see one more question.
Yeah, we want another gene, but maybe we're waiting for that one to finish, and then we can run this one. Okay.
Yes, sure. So just the point is that you can see it here. The recommendation of the system is to run one gene at once. This way it is actually quicker because the analysis are not interfering. So you have to wait for one launch to finish, and then the next one can be launched. So next one we'll do with FBX011 in skin. Okay, so but for this, let's wait for TP53 to finalize. Actually, maybe you have other questions related to the single gene analysis in Match Suite. If so, perhaps it's better to clear them all now before we proceed to the gene set. If you have any questions towards the results that are received, towards the export. And by the way, at the end, we will show you that, of course, the tables that you receive as results can be also visualized from the platform interface, geneXplain platform. And all of you who have access to TRANSFAC and to Match Suite also have access to geneXplain platform. You will notice this product in this perspectives switcher at the top upper corner. And there you would be able to view your Match Suite project, view all the output tables, and work further with them inside the platform. Okay, so TP53 is finished already. This means that we can proceed with the launch of another gene that we just received. And the request was to launch it in skin. Perfect. Okay, let me stay with the default. You'll now see that I promised you to show you in the report that this range is indeed adapted, best supported with no profile optimization. Let's click on next, launch analysis. And in the meantime, while this gene will be calculating, we'll look into the launch results of P53. And just give it a moment, right? So we can go to the view results. I always like starting from the report. As I said, the report is, first of all, you see the promoter range, which was considered, was exactly the one that we have specified. I don't remember. I think that we did specify, right? The tissue, we specified some cortex.
Brain
yes,
cortex.
yes, the cortex tissue, right? So here's the result. Maybe Alexandra can comment on biological relevance of this result. Unfortunately, I'm not a biologist by study, so
Yeah, well, I don't remember those factors.
by heart.
Basically, I see EGR3. It's you remember we had EGR1 last time, but now it's a different member of the family, which seem to be expressed more in brain. So if you click on it, EGR3, sorry. And we can see what this factor is doing. Well, you see in brain and then generally in the tissues like in brain, different brain tissues, as well as in skin, actually, the expression of this factor is quite high. That's why it was selected as a top among top ranking factors, but also the motif seemed to be highly represented in the promoter of that gene. And if you look at it more about this factor, maybe going
okay. Yes, sure.
About involvement of that factor in different diseases. So if you go to the disease biomarkers,
yes.
you see, for instance, stomach neoplasms and bipolar disorder. So we have a relevance to P53 because P53 is quite prevalent in many cancers. And also definitely in these stomach neoplasms, and this factor which is regulating P53 expression seem to be related to this neoplasm. But we also see the bipolar disorder, which is a brain disease. Well, it's a behavior disease, but and we see that this factor is definitely may play a role in regulating P53 in brain.
And glioblastoma, I also see on the list.
Also glioblastoma, that's true. That's
Yes.
the important, cancer in.
Okay, so this is the results. So here we can just comment that there was one enhancer known for that tissue, and in the rest, visually results are similar to what we have seen before, but of course you now have to play around with them to fine-tune them with filters if you want, and to export them and use further for your studies. This will address.
So can we export the report? I guess.
Yes, sure. Sorry, I didn't show. Report, of course, can also be exported, and you have a selection here. It can be either exported as a PDF document. Then it is just a static PDF, like what you would expect from. Let me open it. Any PDF, which would be then hard to be copied in terms of text and images. And by the way, here are how the display tables would look like if we have exported them. So factors, genes, and matrices are all independent tables, which you can then further work with. This is when you apply the filters, when I was showing it to you. But the report can also be exported as an HTML file. And then of course, the text is the text, the images are the images, and it's perhaps easier than to be used for, I don't know, publication purposes or whatever you need the report for. So you can, or you can, of course, make the screenshots from the interactive interface, depending on what you need. For instance, genome browser visualization for sure can be screenshotted from the results overview. Okay, so that was the P53 run in the cortex tissue. This one is also ready. Okay, let's have a look at it. Again, perhaps maybe let's just start from the report. By the way, as you have noticed, there were no enhancers known for this gene in the skin tissue, I believe. This is what we selected, yes, the skin tissue. So here are the transcription factors.
Yeah, it seems
table.
we have like AP2 is quite specific for skin, but it's number 2 among expressions you see. If you look.
And then the third one is
so that's AP2, gamma and alpha. They both are quite specific for skin. Although AP2 is quite a general factor, but still it has a certain priority in skin.
Actually, no, Alexander, since we see it in red, let's have a look. Since we see it in red, it should not be actually a general factor. Let's check. But that means that actually it's when you see something closer to red, its expression profile should be very, you see? Actually, you cannot say that it's evenly distributed.
Yeah, skin is number 2 and the first is placenta. But yeah, skin it's also quite specific. That's true. And you see down in the list what I see also KLAF4, and that's something we'll see today because I know what is Daria is going to present today. And again, it's playing quite a good role in skin development. But we'll talk about it a bit later about this.
Perfect. Okay, so just to make sure, if there are no further questions for single gene analysis in Match Suite, we can proceed to the gene
by the way,
analysis.
so the person who asked us about this gene or both genes, if you like, we can send you these reports. Just I guess we will be able to find out who.
Yes, you can send us in private messages, just in the private mode, your emails, and then we will send you the reports. Indeed, the PDF versions you for sure can open, and we can also. But okay, if you have TRANSFAC, as you saw, it takes only a minute to regenerate it. I mean, in the system, the launch is very quick. Otherwise, we will send you the analysis reports as PDF docs, and you can make use of them for your further studies. Okay, perfect. So just please make sure that you send us in the private mode your email addresses, and that we have them. Okay. So as I understand, there are no further questions. Yes, so now we can proceed to the gene set analysis. But for gene set analysis, there are also several options to launch it. Okay, since we are now in the interface of the Match Suite itself, not on the portal interface, let's start here. So once you have selected the gene set analysis option, click on next. You see there are quite a number. Okay, I see another questions. Okay.
No, this
Yes.
emails.
So you have several options. First of all, of course, we were so nice to compile a demo gene list, so you do not have to bother at all about what the input should be if you just want to test the system. Then you can use the demo gene list for your run. You can visualize it. So to be honest, I do not remember how many genes there are in it. So 50 genes. I believe that they were selected from some experiment just by look full change filter, but to be honest, I do not remember, so I will not be insisting on that. Anyway, there's a demo gene list you can apply and launch the analysis on it. Or you can use your own data, of course, and the supported input formats are gene IDs, either in gene symbol, in ensemble ID, or in trace ID. So just a column of your genes with respective IDs should be then inputted, or you can actually even copy paste them. So just copy paste them here in this window, and this will be saved as a gene list in the system, or you can upload the file or the browse stored data, especially if you're a platform user, you have some results in the platform where there are gene lists inside, you can just use those files. And I have my saved data here, so I have bone development list and skin development list. In a moment, you'll see how they were actually constructed, but not now. So for the beginning, we can just proceed with the demo gene list. Go further. You have to specify the name of the launch. Again, you're selecting from the same 61 tissues that are currently supported by Match Suite. You can specify the promoter range, as you know. And this is very interesting. You can optimize your gene set. I mean, you can have selected your genes from indeed some experiment, and maybe you don't have time to investigate what are actually those genes doing and whether you're interested in all of them. And if so, you can select this option of gene set optimization, and your input gene list will be optimized by the geo categories, which will be found to be enriched in your input genes. So for instance, if I select biological processes and click on next, give system a while to work on it. Currently, functional classification is being performed on the backend. And once this functional classification will be calculated, we will see a kind of tree map with respective biological processes that were found to be characterizing our input gene set. Okay, here it is.
Daria,
yes.
Maybe in between you can answer this question.
Sorry.
Is it free to use, or we need to subscribe for the software?
Yes. So Match Suite is a component of TRANSFAC. So you need to subscribe to TRANSFAC and then any type of TRANSFAC. So on our website, we will show it at the very end. You'll find a variety of TRANSFAC packages. Any TRANSFAC package would include Match Suite. So you can make use of it from any of TRANSFAC packages. But yes, you need to be subscribed to any TRANSFAC license to have access to it. Okay, so now you have this geo map. As you see, it's interactive. To be honest, I'm not a biologist, as I said, but I can select, like say, double click selects the whole category, and a single click would select the subcategory, and you immediately see the number of genes increasing here, depending on the category that I used. Sorry, my selection was random without any biological sense, but I have made it to more than 20 genes, which is the minimum limit to launch the analysis. And with this selection, I can proceed with the selected genes. And then only genes belonging to those geo terms from my initial input gene list would be taken into the analysis. And you can actually have a look at how this list was optimized. So before we had 50 genes in that list, as you remember, and now there are 35 because only 35 of them belong to these categories. And now we can launch analysis. So I will again click on launch analysis, but I will immediately stop it, not to interfere with the demonstration of the ready results. If single gene analysis is indeed taking just literally a minute, you have seen it already. Because we were running live the genes of your interest, the gene set analysis takes a bit more. but it's just also several minutes, so it's not that you have to wait for hours for it to complete. Similar to single gene inputs, you can see here the summary of the input parameters that were used for the launch. If any geo categories were used, you can also see them. And the input is clickable, so it will again reopen you the gene list that you have run the analysis on. But you always recommend to carefully name your analysis so that you do not have A, B, C, D, E, F, G, but it is something meaningful. And then already from the launches history, you know what was the input just because you named the analysis in similar way. So as you see here, in my launches history, I have the demo gene list, which was run without any additional restrictions, so without tissue selection, without geocategories selection, and I would like to show you the results. So this is the most generalized view of the results. It will be talking about expression in average tissue, so average levels of expression across all supported tissues for those genes. And again, similar to how we were doing it with the single gene analysis prior to jumping into the interactive interface, I would like to start from the report, which again gives you the summary of who launched the analysis, when, on which release of the database, what was the input gene list used for the input. And now you immediately see the summary of transcription factors. And you see that at least 42 genes from the input gene list, which contain 50 genes, are regulated by these transcription factors. And below you will see how. But prior to jumping into these results, again, I would like to first give you an idea of how the analysis process looks like, so you're well prepared for the discussion of these tables that we got as a result. So for this, I have another slide. And again, just to remind you, of course, here in the report, you also have this overview of these processes that we will be now looking at. And of course, you have the links to the methods. I think, yes, from the very beginning, you just click on these methods and everything is described absolutely in all details, whether you have applied any additional conditions, how they will be used, and so everything will be in this method section. So now we're going to just briefly look at the analysis pipeline. So we start from the selected input gene list, and we run so-called fmatch analysis on it. Fmatch is checking for enrichment of binding sites, and for this, it also needs a control set. These control sets are randomly sampled. So first of all, sorry, what is the track that we have inputted to this fmatch? Of course, these are the regulatory regions of the input genes. So these are promoters of the genes from our input list. And now we have to have some control set. So these promoters are all composed into one track. It is a yes set in our case. And now we need the no set. And these are randomly sampled promoters from other regions of genome that do not intersect with our yes set. And we are running this 5 times. And eventually, we're now actually constructing the profile that will be further used for the analysis. And eventually, for these matrices that we will be finding as a result, we will take only those that none out of these 5 runs accidentally got also into the no set. So it is very strict. They should not be in the no set. So because we want them to be characterizing only the yes set and with the high level of reliability. Then comes our favorite step. It is absolutely the same as we discussed before. This orthologous and paralogous extension is when we make the predictions actually even wider. But then luckily, the redundancy filter comes, and here, in case of the gene set analysis, for each factor cluster, we leave only one matrix, which is maximizing the adjusted site enrichment value from the results of the fmatch. Again, fmatch is a clickable method in the geneXplain platform. It's actually called search for enriched TFBS on tracks. If you want to find it, of course, it's linked to the methods section is Mesh Suite. You can click and immediately open it in the platform, read its description, read related publications. Everything is interlinked there. So then we are filtering we are filtering those matrices in respect to the obtained results. and we are optimizing basically this profile that we are constructing. And then having constructed this profile, again, a customized profile. So here we use, as you remember, 5,700 something matrices. Here is much less. It's an optimized profile, which is actually now used for the site search. And it is used for running the match, the classical match algorithm, and the composite model analyst algorithm inside the platform. If any of you have attended our previous coffee breaks, Alexandra was talking a lot about general site search and about the combinatorial analysis of transcription factor binding sites. There are even independent coffee breaks related to this topic. So this is exactly the CMA. It's the combinatorial analysis. And this is just the site search method from the platform. And having put together those results, we're getting the eventual list of matrices, which is then mapped to the transcription factors corresponding to these matrices. And those factors are again ranked to select the best ones. And here are the parameters of how they are ranked. So it should be the maximum value of adjusted site enrichment for the factors matrix. It should be presence in the constructed CMA model. It's not should, it's just ranks. And then you take the rank sum. So those which got it into the composite model have advantage. Those that have maximum values of adjusted site enrichment have advantage. Those that are highly expressed in selected tissue or average across all tissues, or those that have a good rank of factor expression in selected tissue or average factor expression rank is high, also get advantage. And having applied the rank sum in respect to all those values, you get the resulting list of transcription factors. And exactly this resulting list of transcription factors is what you see at the very top of this report. So, right. Maybe let's go through the whole report and then we go to the interface, and there it will be even more obvious how this values can be used and constructed. Just maybe to comment a bit. So this is the so-called simplified factor view. So okay, just factor view. This is exactly the same table. Then there's also Factor View Pro. In the Factor View Pro, which I think is not making it to the report, you see more details, for instance, about the factor enrichment. And here this enrichment is just reflected by, the color of the dot. So if you see a blue dot, that means that the enrichment of a respective site was higher than 1. And if you see a green dot, that means that in addition to that, the sequence enrichment false discovery rate was lower than the threshold 0.01. So site enrichment, I think it's obvious, right? Is when you have a lot of sites in the yes set when you compare it to the no set. And sequence enrichment is meaning that sequence are the promoters, input promoter, that you should have at least one site of this matrix in the promoters of the genes you're studying. So this is what this color is reflecting. This combinatorial analysis tick, if it is there in place, it means that the respective matrix made it to the combinatorial module of CMA. Actually, in the methods section, we describe in details how these models are constructed. And as I said, Alexandra even had an independent 1-hour coffee break devoted to that. But maybe just to make it very short, it is a genetic algorithm running. Alexandra, you can correct me if I'm wrong, but it is okay.
Yeah, just go ahead.
Okay,
yeah.
This is a genetic algorithm running at the back end, trying to find such combinations of transcription factors that separate in the best way the yes and the no sets. So this separation point actually is what you see here. And so each time it is fine-tuning the parameters of this separation to trying to make them separate the yes and no sets as much as possible. So this is to make it very quick. And to make it not quick, you actually see all the descriptions here, the sliding windows, the formulas, and of course it is all linked to the publication where it is described. So and as I said, we had an independent 1-hour long coffee break where Alexander is talking about this algorithm. You can of course find it on our YouTube channel and on our website. So the tick here means basically that the matrix made it to the module of the composite module of CMA output. Okay, and then we are already prepared for this. We know that we're taking expression values into account. And since we have not specified any particular tissue, we just request the factor to be, in principle, highly expressed on average. That's why this values here are talking about the average expression and average specificity. And again, the color means the same. That if it's red, then it's specific. If it's green, it's not specific. So this is very similar to what we had. So here's this scale of colors. Sorry, starting not from green, but even from blue. Here you can see it. Okay. Now, this is something interesting. This is, and we will see it also in the results visualization here in the genome browser. Since we were performing actually two different site searches, right? We found the enriched sites, and we found the CMA. Yes, so now you see them. So we have sites that were predicted by the genetic algorithm. This is from the CMA model, right? And we have sites that were predicted to be enriched when we compared the yes and no sets. And basically, that's what you see here in this report for each of these are promoter models for all genes from our input set. And they are actually sorted, I believe they are, by CMA score. Yes, you see it is decreasing, this number here. So actually, the CMA score tells you how well this model is characterizing the promoter of this gene in contrast to the promoters from the background set, from the no set. It is telling you how good this model is able to identify that this module of matrices of transcription factor binding sites is really regulating our target genes and not some other gene from the no set. This is what this value is reflecting. And so we're sorting by it. And so, of course, we're not giving it for all input genes. I think we had 35 for this run, but each table has view full table mode. And if you click on it, these models will be constructed for absolutely all genes from your input set, and you can browse and visualize them. And of course, the lower we get, the less is the number of sites found, and the less is the score. Okay, now we already went through the schema while talking about how we obtained the tables. Yeah, so you're well aware of all the steps, which are again all clickable with all the supplementary materials, methods, links, publications. Here is the visualization of the composite model that was constructed for our case, and matrices that have the green ticks are from those modules. And we'll see it on the interface. This is how the yes and no sets are separated. So actually it's very good false negative and false positive rates. So we have a well defined separation point. And now you have the factor table. Oh, okay, it is. So before we had only with this green and blue dot, and here instead of the dot, you actually see the values and you see the factor rank. But it is basically the same table as at the top, just with additional information provided. And then you see the summary table of matrices. But as we know, we first construct the matrix table, and based on that, we then select the factors and rank them by characteristics that we had on the slide. So it's just in the report, the matrix table is below. And then we eventually see the full model of how our genes, so here are all genes from our input list, how they are regulated, by which transcription factors belonging to which matrices, how much sites were found, what was the CMA score of the promoter of respective gene. And now we are ready to go to the online visualization, because this is exactly what you see here. So this table we have been just looking at is here. Here are our genes. Again, the intensity of the color means the number of respective binding sites found on the intersection. Here are the tables of factors in two different views. The brief one from the very top of report and with some details from the very bottom of report that we have just saw, and the matrix table. And the genome browser, of course, with the results. And to view the sites predicted for promoter of each particular gene, you just click on that gene and the picture below is changing. Now, as you might notice, we also have filtering options here. And they are actually very interesting because in case of gene set, here we did not specify the tissue for this launch, as you remember. We just launched it on without specifying a tissue. But now I might have decided that, okay, in reality, I am not interested on all binding sites that were predicted. I want to filter and see only those of them, at least one transcription factor of which is known to be expressed, I don't know, in bone marrow, for instance. I select this and click on Apply Changes, and my already calculated results from my run are now being filtered. And we will now completely exclude all matrices, and so respective binding models and transcription factors referring to them in our prediction, which the genes of which transcription factors are not known to be expressed in bone marrow. So now we are adding additional filter by tissue right on the fly. Of course, it takes a while because it's basically recalculating all results for us. And now it has finished. And I didn't pay your attention to the total number of sites that we had at the beginning, but I will do it now when I'll remove all filters. And you'll see that for sure, these numbers decrease. And of course, the visualization on Genome browser gets smaller, and you are not restricted to selecting just one tissue here. Actually, on top, I'm not sure if there's any biological relevance in that, but in addition to bone marrow, okay, whatever, I can ask it to be also expressed in esophagus and apply the changes. And now just pay attention to these numbers, the total number of sites, they of course will decrease once again, because we're cutting the number of matrices even more.
Yeah, it's actually an interesting way of comparing, because as you remember, the initial search was done on a whole set of matrices, which are without selection for a particular tissue, although this set of genes, they give us a restriction. So we only choose those matrices which gives the best presence in the yes set compared to the no sets. Now we want to look whether found factors, whether they expressed in different tissues, and whether the respective sites can be also considered. So that's different from running the same from the very beginning. So you would select the tissue from the very beginning, and I guess you will show it in the
yes,
next
of course, in a moment.
And then in such case, the tissue expression will be involved from the very beginning in selecting the particular matrices to use. So it's a little bit different approach here, but
this is really for filtering. This is really to fine-tune your results. This is like, so as Alexander said correctly, if you select the tissue from the very beginning, that affects the whole analysis process. That affects the promoters that we take, right? Because we take them from Phantom 5.
Also can be specific.
Yes, it affects the ranking of factors, whether they're selected for the eventual results or not at all, and so on. And here we're just talking about filtering of the already calculated results. But it is a very interesting type of filtering. You can really fine-tune them, and you can actually see, maybe you do not expect, so sometimes maybe you can make interesting investigations with such filtering, which it takes, just as you saw, just a couple of seconds, and you might play around with applying different filters by tissues of your interest, and you see immediately the values that were calculated for those tissues, and you can sort, of course. I mean, you can find those which are having the bigger the difference from average and the bigger levels of expression in this tissue in principle. And if you understand that you have lost the initial filtering by the best factors, you always have these best factors on top, and you come back to the initial sorting with the best factors on top. Okay, I can now remove all filters. Just give it a moment to return to the initial state. This was the factor pro view with the tissue filtering, and we also have the matrix view. And the matrix view can be filtered by two parameters, by adjusted site enrichment and adjusted sequence enrichment. So, so now we have 27 entries. If I apply the filter, I immediately have 20. Or if I apply that one, I have 17. So I play around, I look at the results. So this really needs your time to, you can of course extend this table to see it all at once, to play around with the filters. And once you like what you see here in this window, you click on the Apply Changes button. And now here, our results will be recalculated because matrices that did not make it to these filters, and of course the associated sites and factors, will be excluded from the tables and from the genome browser visualization. Just give it a moment. Yes. So this is how the filtering is applied. And again, you can download the tables. Once you see that you applied the filters in the way that you like them, you download the tables and you save this result in such a way. So you can jump from promoter to promoter and visualize what you have in the genome browser. Genome browser might look similar to you, right? You again can click on any site, find this information, go to the database, look at the respective entry, but it actually has other tracks which are displayed, and maybe let's talk about it a bit more. So you can visualize just by clicking on it, they will be added or removed from the view. All genes, promoters, and enhancers, the genes track, which is already there, all enhancers, all variations. That's interesting, by the way. So if you're, for whatever reason, interested in the variations track, okay, maybe not here, but they will be, okay, like here on this promoter, you see there are a lot of variations, and you can click on it and you can see the alternative allele, the reference allele, the name of RS ID of respective variation. If for whatever reason, for instance, you're studying how your transcription factor binding sites can be affected for by variations, and you feel that some of these variations can be relevant to your research, this is one way to look at that. Then you have experimentally validated transcription factor binding sites. It's the same as we were doing it when the single gene analysis. You just see independent track of those that were validated, and then you can just visually compare which of the predictions were already shown in an experiment and which not. Then conservative regions of genome is also the same as from the single gene analysis. Track of all found sites, and then track of promoters, of enriched sites, and CMA sites. So enriched sites and CMA sites is what made it to the promoter model in the report, to these nice images. But here, of course, using all these ticks, you can adjust the way you visualize it in genome browser, and you can jump to various regions of the promoter regions of your input genes and make out nice screenshots from that. In addition to just visualizing the tracks in genome browser, you also have ability to filter by them. For instance, you can only view those sites that are intersecting with enhancer regions, or you can view only those sites that appear to be near the conservative regions of the genome. This is similar to the filter we already applied for single gene analysis. But anyway, let's first apply that filter. And please note that the table on top will be updated respectively. You see, you immediately got the zeros. Oh, okay, so it's 6, it's not 0, 0, 0, so it's 2, 1. So it's not much sites that are located near these conservative regions, and we can view this track, of course. Yeah, and so that's why we see only those sites which are under the conservative region. That's obvious from visualization. Maybe let me remove those SNPs so we do not move attention to them. And another filter which you can apply is view sites intersecting with enhancer regions, if any. Let's see. Okay, so I see that, okay, this one got, okay, only one got it. And yes, so we should view the enhancer tracks for that. Yes,
all
right.
enhancers,
Sorry. Yes, I selected the wrong
then
one.
you can maybe yes, you can see.
Yes, exactly. Yeah, so this is in that one enhancer. Okay, perfect. And yeah, and by the way, it's clickable and it will take you to the respective enhancer report in the database. So whatever you click on will have additional information and will lead you to the respective information in the knowledge base. Okay, that's in general how the results of GenSet analysis look like. But just to remind you, we made the launch without specifying any interesting conditions, right? So I have an alternative launch where I have used the same demo gene list, but I also applied the lung tissue And I was interested in gene ontology categories of signaling and system development. This is from interactive tree map that we saw. This is just quickly to show you, of course, you do not remember by heart what we had in the previous results, but these results would be different. And since it is interactive, for instance, the here in combinatorial analysis, by the way, none of them made it to the very top 10, I believe, but we can view more, maybe in top 25. Yes, we have some. Or another way would be to, again, go back to the view of 10 and just sort them by combinatorial analysis. I'm just giving you an idea how to work in this interface. The same for expression filtering and the difference from average expression. So the results are very similar from technical point of view, but as I said, of course, they're absolutely different from the point of view of found and predicted sites. And another example, which I would like to show you, of course, you can also load your own gene list, as we saw it from the very beginning in start analysis and GeneSat. You can upload your own file with ensemble IDs and TRAS IDs or gene symbols. But there's another way to jump into the analysis of Match Suite, and it can be done from the interface of our portal. And I find this actually very exciting. Well, if you're not searching for one particular gene, but instead searching for some gene set, there's a very nice function here, which is called ontology search. And in this ontology search, again, depending on your license, you can search for diseases and drugs if you're also subscribed to human PSD and TransFac. But for instance, this geo-ontology is available to everyone who has TransFac. And in this geo-ontology, you can search, for instance, here in this field, because I'm not very well aware of how gene ontology is organized, to be honest. I can just search for skin, and I immediately find a number of results. And here in this results, for instance, I can find skin development. And the system immediately lets me know that it knows 450 genes assigned to the skin development category. If I was smarter, I could have found them by iteratively clicking through the geo. So I would have known that it's development process, anatomical structure development, system development, animal organ development, and finally skin development. But unfortunately, I'm not that well aware of gene ontology classification, so I apply the search. So I view those 450 genes assigned to this category here in the BKL interface. Here they are. By default, I think I saw top 25. And now there are two ways to proceed. As you can see, the species here is a mixture. It's mouse, human, I'm sure there are others below. So what I can do is actually I can apply a filter manually. And here I can specify the species taxon and say that it should be human. But I will not, because Match Suite is very smart. Even if I select all those genes, all 450 from absolutely different organisms, mark them all, and click on Match Suite gene set here, the system will automatically, for me, check which of these genes are actually human genes, and it will proceed with the analysis of only the human genes. So out of this 450, I will not even, okay, let me name it like skin 2, just to differentiate. We'll see how many genes we actually eventually get. So again, in this skin, I can, of course, specify that I'm studying skin tissue. If I want, I can narrow my gene list by GO terms, as we saw it at the beginning, but I will not do that now. And here I can see that out of this 450, 199 genes actually were human genes and were taken further to the analysis. But okay, so now I could have clicked on launch analysis, but I have, of course, done it already for us to look at. And here in my launch history, it is called the skin development. Just for you to know that it was exactly this skin development, 199 genes, no narrowing to geo categories, and skin was specified as the tissue in focus. We can view the results. And since we're now actually in a quite biologically relevant context, right? We're looking at skin development genes in the context of skin tissue. These results, we expect them to look biologically relevant. And here I give floor to Alexander to comment on their biological relevance.
So, okay, so if you return back to visualization first,
yes,
By the way, you see EGFR, which was in the focus on
Yes,
the
it's the
beginning.
first gene.
Today is the one of the first genes here, so it's obviously it is expressed in skin, and actually it plays a big role in skin. And if you look at the genome browser below, then of course you see different sites compared to what we found when we were analyzing this promoter alone. And that's important to understand that depending on the condition. So here, the set of genes which we are starting. So we are starting with a set of skin genes. And this set, these promoters of skin genes, they're creating, they're actually defining the condition. So we are searching for those motifs which are enriched in this set of promoters. So, and which means here our focus really on the transcription factors and the motif, which are first of all expressed in skins at all, but also they are specific for that particular subset of promoters we are analyzing. So in that sense, you may see different factors and different sites compared to what we found when we analyzed just one gene. That's first thing that I can immediately see here, but also looking at the transcription factors. We already we're talking about KLF4, and in this case, it's number 1 in the list. And if you click on it, you will see information about this gene. And actually, what you can see here is really, information about where this factor is expressed, and you see clearly skin and stomach, and you also see here immediately that in skin it's number 1 in terms of expression. But what is also important to understand that this is not simply expressed in skin, but playing really a good role. So I send you, Daria, in the chat the link to a paper which is a recent Nature paper. Well, not really recent, but it's very good Nature paper, which tells that KLF4 is a transcription factor required for establishing the barrier function of the skin. They actually telling that this is very early in development, so it's really skill development, and it's playing a role on the early stages and also in the whole life when the barrier is maintained. So when as you know, skin is always changing. The epithelial cells are always changing, and the old cells are dying, but new cells are developing, and this function of the barrier function, that no intruder will come in, are always maintained, and KLF4 is really a key factor playing the role in that skin development. So in that sense, our results of just analysis like that, so really came up as factors at the top of the list, which indeed playing an important role in skill development.
Great. Thank you very much, Alexander. So I'm not sure if you would like to provide further comments. In principle, results technically are always very similar from brief glance. But of course, once you start digging into the details, once you start applying the filters, once you start fine-tuning the parameters, you immediately get various outputs, and you need to spend time understanding how to make further use of them. And this will briefly discuss in the next session, a section of this demo devoted to geneXplain platform, really briefly, but just to give you an idea of what can be done. But maybe one last thing I would like to show. For start analysis, in case of gene set analysis, you can also start from constructing a tissue-specific gene list, as it's called here, from scratch, if you have subscription to Human PSD. What that means, for instance, you do not have a gene list in mind, but you know that you're working with some particular tissue, for breast, for instance. You have to wait. Now the system is doing the following. It takes the 10% genes, the top genes that are known to be expressed in breast in principle. So of course you do not see here all thousands of genes, but you see the selected top. And here with your pointer, you can select a window of the genes which will be selected for the analysis. Here, you see the level of specificity, meaning that those genes are very specific for breast tissue. So the higher they are, the more specific they are. And the lower they are, the more general expression level of this gene is. Expression level is shown here as an absolute value, and here is the specificity. So expression by absolute values from protein atlas and calculated tissue specificity by us. All these ranks that we saw is exactly the specificity, and the color, like red, green, and so on. And now you can adjust this window of genes that you want to select. You have to have minimum of 20 to proceed with the analysis. But just to show you that this is another way of starting analysis in match suite, is just starting from the tissue type and then proceeding with the selected genes.
Right.
Okay, I think that from my side, that was all that I wanted to show. And now hopefully we can have questions or just open discussion for the gene set analysis. And then we will briefly show you what can be done in the geneXplain platform and what it is actually at all.
Yeah, right. So don't be shy, just to ask anything you like. For instance, one point we maybe was not very clearly explained yet, but when we select a tissue, it's not only selection of factors which are expressed in the tissue, but it's also, which is very important, a selection of a promoter, which is a tissue specific. So as you know, the same gene can have several promoters and several enhancers, as we have seen, but in a particular tissue, only one promoter is acting, or and this information is taken from various sources. So the main source is Phantom 5. It's a database where extensively all human genes were also mouse, but we don't have them here in the system. But in Phantom 5 for human genes, really position of start of transcription was mapped in different tissue types, and that's all information is loaded in our system. And then when you select your skin tissue, for instance, for if this gene has a skin specific promoter, we're looking for that promoter.
Yes, thank you, Alexander. Maybe just to summarize, tissue is playing important role on when you select the promoter, basically the studied region, when we optimize the profile that would be used for the site search. So we require matrices to have at least one connected transcription factor, whose gene is known to be expressed in this tissue. Otherwise, it cannot be acting there. And it is also used for ranking, eventual ranking of the resulting transcription factors. So when we already came up with a table of matrices, we now need to construct the factor table based on it. And those factors selection for this table is made based on the expression of values and other different parameters that we have calculated based on the tissue expression values. Okay, since we do not have any questions so far, maybe we can briefly proceed to the geneXplain platform. Just to give you an idea of what it is, of course, we do not have time today. It was already 2 hours now that we were talking. Just to give you really a brief idea, to come up with a gene list, for instance, you might have a gene list of your interest already, or you might have raw data, like raw transcriptomics data, for instance. Then the platform can be used for identification of differential expressed genes. You just click on RNA-seq tab, you find how to load the data, you find how to detect differential expressed genes, and the platform allows you to process your raw FASTQ files and to come up eventually log fold change values, and then based on them, you can make some selection of genes and submit this gene list to the match suite as an example. Or perhaps you can process your match suite results further, and then you can do perhaps pathway analysis based on the transcription factors that were predicted to be regulating the gene set of your interest. Actually, the platform is very advantageous because it has integrated promoter and pathway analysis in one workflow. So basically, this workflow allows you to start from the raw data and come up with the list of master regulators responsible for regulation of genes referring to the input set, but then you do not even need to apply mesh suite. We also have another system, Genome Enhancer, that is working with human data as well. And in case of Genome Enhancer, you do not have to take care at all about selection of which methods and which workflows to launch in which consequence. You just open the system, upload your raw data, and it immediately will take you to the results. For instance, starting from raw FASTQ files to the ready report with identified master regulators after performing promoter and pathway analysis. So we have a whole world behind the system. And maybe just to give you an idea, so here in the platform and the left tab, which you open by this menu button, you see all databases available to you. We have free databases such as GeneWays for pathways, GTRD and Hokomoko for site analysis. We have the data tab where you'll find example data. And by the way, I believe that here under the question mark, yes, you can also find some exercises and of course a user guide and a quick start guide. And the sample data can be used for that. Then we have projects where you will see your own projects together with the demo projects available to you. Those that would be your own would be then marked with a green read-write access, and those with red means that these are demo projects in the platform. And you have a public data folder. And of course, the main tab is the analysis tab, where you can then find all the methods that the platform suggests. So if here it is just the brief summary of what you might want to get, let's call it frequently used functions, but here it is the full tree of what is available. We have Galaxy integrated. We have a lot of methods for just manipulating the data, for normalizing, for functional classification, for instance, ontology annotation, site analysis, of course, and so on, a lot of stuff, and the workflows. Workflows are based on the respective database. For instance, for TRANSFAC, you expect to find a lot of workflows for various ways to identify transcription factor binding sites. And if we're talking about further steps of analysis of match suite results, you might want to have a look at the transpath workflows for finding, for instance, the master regulators based on those predicted factors. Okay, this was very briefly about the platform. If we do not have any questions from my side, I think I've told you everything I planned.
Thank you very much, Daria. It was very intensive, but I guess it was helpful. Yeah, these 2 hours just ran very quickly. I was very amused, but I hope you also for listening us. So far we don't have any further questions, but I think you will get questions. If you have questions, just send it to us by email or by the form which we have in this announcement. Okay, thank you very much, and hopefully see you next time, and have a good day.
Thank you very much. Bye-bye.
Goodbye.